And now let us verify the biochemical reactions involved in CAM pathway. And total dark phase mechanism in CAM plants occur in mesophyll cells only. There is no physical separation. All the reactions occur in the mesophyll cells only, but some reactions followed in night as well as the other reactions that occur in daytime here we can follow. And as we see the reactions that occur during night time in CAM plants, and that is formation of malic acid, that is acidification, and here the primary acceptor is phosphoenol pyruvate catalyzed by pepcage we can see. And so this is the primary fixation of carbon dioxide and in the form of malic acid, that is stabilized during night time, and during daytime that malic acid undergo decarboxylation here, and that carbon dioxide evolved during daytime in the mesophyll cells in presence of assimilatory power successfully reduced to carbohydrate through Calvin cycle pathway or C3 cycle. And the pyruvic acid now converted into phosphoenol pyruvate again and regenerated. So as we follow these biochemical reactions, this biochemical pathway is similar to that of C4 plants. What we followed in C4 mechanism during dark phase, and in the same way here also phosphoenol pyruvate fixes carbon dioxide initially in the form of 4-carbon organic acid malic acid, and later that malic acid undergo decarboxylation and supply carbon dioxide to Calvin cycle, which is a similar process we observed in C4 plants. But there is a difference as we compare C4 pathway in C4 plants and CAM pathway in CAM plants, that mean in succulent xerophytes, and whatever the changes that occurred in the mesophyll area in C4 plants and that occur during night time in CAM plants, of course in the mesophyll. But as we verify that second fixation, that ultimate fixation and Calvin cycle reactions and deacidification, that is decarboxylation of malic acid, such events and the same events that we observed in C4 plants also, but occur in bundle sheath cells, and whereas in CAM plants and these reactions occur in mesophyll cells only but during daytime. So as we compare that C4 mechanism, which is meant for alternate pathway for CO2 fixation, and CAM mechanism, which is to support that scotoactive stomata in succulent xerophytes, and the biochemical reactions are similar, but they differ in space and time. In C4 plants they differ by space, occur in mesophyll and bundle sheath cells, but whereas in CAM plants they differ in time. And the changes that occur during night time and during daytime here we observed, but in the same compartment, that is mesophyll. And that is the difference between CAM plants and C4 plants, and both maintain that alternate pathway for CO2 fixation, and they followed that acidification and deacidification processes, and that is in order to supply CO2 efficiently for Calvin cycle reactions. Okay? And that is about C4 pathway and CAM pathway, and we discussed about C4 plants and CAM plants in detail. Students, now let us follow a simple numerical based comparison, and that is comparison of energetics utilized for dark phase mechanism in C3 plants and in C4 and CAM plants. And there is a variation. In C3 plants only C3 cycle takes place, that is for both fixation as well as reduction. And for that fixation and reduction of 6 CO2 molecules into 1 hexose, already we calculated 18 ATP and 12 NADPH2 are utilized. That is the assimilatory power required for the synthesis of 1 hexose in C3 plants, as they perform only Calvin cycle reactions and both fixation and reduction occur in Calvin cycle. Okay? But whereas in C4 and CAM plants, as we followed, they perform the alternate pathway for CO2 fixation as well as the usual Calvin cycle reactions for the reduction of that carbon dioxide. So they perform that C4 cycle, that is C4 pathway, as well as C3 cycle.